thank the staff and everybody here for inviting me. I've been asked to talk about a interesting topic, something that's been dear to my heart for many years, and that's about the molecular and biologic basis. This is sort of a setup for potential therapies that I think I'll discuss a little bit in the afternoon, because unless you really understand some of the basic pathomechanisms, it's hard to devise treatments. I thought 20 years ago when I entered this field that I could just blindly, like a typical orthopedic surgeon, shoot something into the disc and make things better and then make it normal again. But it's never that simple. And as Dr. Eyre pointed out, even the intricacies of collagen is so complicated that we may have an uphill battle. So the disc has been researched over the last 50, 60 years. Really, some of the early publications started in the 30s and 40s, and in the JBJS literature and anatomy literature, really hasn't been around too long, and people started understanding it a little bit better. But there's a whole time period where the disc was pretty much ignored because it was thought it was just a gristly substance, it just degenerated and nothing can happen. Very similar to what people thought of cartilage 40, 50 years ago. But as it turns out, the disc and cartilage have many things in common, and some of the biologic mechanisms are very similar. So when you look at a normal disc, that's a beautiful structure and a lot of intricate details about it. So I'm going to go over some of the biological, histological, and molecular basis of why this turns into something like that. And Dr. Eyre pointed out there's genetic predisposition, but even if there is a genetic predisposition, why only one disc degenerates in someone that has same genetic issues at the other disc levels? So it's not only genetics, probably is a significant component, but there's probably epigenetics, gene-environment interactions, and why only one of the discs may degenerate, and in another patient where disc can actually prolapse and cause other neurologic problems. So what is it? I mean, we've gone through this. It's basically a structure that modifies the forces transmitted between one vertebral body to the other, and it undergoes a lot of mechanical loading, as Randy pointed out. And it's also an extremely intricate structure. It's not just disc annulus. There's cartilage end plate, there's bony end plate, so there's a lot of complicated biology going on here. And suffice it to say, I think we still do not really have a true understanding of what's happening here. So, as it's already been pointed out, this is just general review. There's a lot of matrix components. In the center of the nucleus, we know there's a lot of type 2 collagen. It's the most abundant in the nucleus, and in outer annulus, there's type 1 collagen. And then the matrix proteoglycans in the nucleus is what gives it that glistening jelly-like structure. So there's a general order to how these matrix proteins are lined up. And if you carefully break down these matrix molecules, you will find that one of the most important, other than the collagen, I would agree with Dr. Eyre that collagen is extremely important because this is what the proteoglycans attach itself to, and probably the foundation of how the whole matrix biology works. But on the other hand, for those involved in matrix biology, some would argue that this big old aggrecan protein is very, very important in what happens to the disc. It's a very, very complicated structure. It's a big hyaluronic acid mixed in with the core proteins that attach these gags. So it's a very, very delicate but very interesting structure. And it comprises major portions of the matrix and the nucleus pulposus. Interestingly, there's a lot of packaging that has to get done, and you've heard again from Dr. Eyre that there's a lot of collagens that are involved, and these matrix proteins have to assemble themselves. And I must say, it's a bit of a mystery how all these matrix proteins assemble themselves, because these cells also produce all these other various type collagens, including type 9, as been previously mentioned. And there's also other matrix proteins like elastin that gives it some degree of tensile strength to this annulus. And as the architecture is put together so beautifully out in the annulus, one would think that in the nucleus it's all disorganized, but people have actually shown that these collagens are actually You would think that it's just a haphazard collection of collagen, but actually these type 2 collagen fibers go from one end to the other and attach, but there just happen to be long strands that mingle together. So it's actually not as disorganized as the nucleus appears. So basically, from a mechanical standpoint, when you have a normal annulus that's well confined, it's like a radial tire with the nuclear jelly-like component in the middle that imbibes the water due to the strongly negative charge of the proteoglycans. And in essence, this is what gives the disc its strong biomechanical characteristic. Now, all matrix has to be maintained, and typically the cells within the matrix maintain it. We all know that. In the human disc, during childhood, the neurocortical cells seemingly disappear. That's in debate right now. Some people argue that neurocortical cells may still float around in there, but in a little bit of a different phenotypic characteristic. The nucleus has chondrogenic type of cells, and the annulus probably also has cells that are chondrogenic in origin, but behave like they're fibrous tissue or cells. But wherever the cells are, they put together this well-orchestrated matrix biology, whereby once these proteins are all made and they get dumped out into the extracellular space, somehow there's an orchestrated packaging of these proteins into this complicated matrix. So that biology itself is a bit of a mystery as to how this is all put together. There's all these other comp proteins and other things that the cells make and then dump out into the extracellular matrix, and how it's controlled, that no one really has a clear understanding, even if you talk to the proteoglycan biologist. So I'm telling you that this is obviously very complicated, and it's never that simple. But, when you start looking at the disc tissue itself from embryologic development, we all start from an amorphous glob of cartilage, and the disc cells and the notochordal crest actually involutes and becomes the nucleus pulposus, and then even the bony end plates and the cartilage end plates initially started off as chondrocytes. And a word about the notochordal cells. There's more and more disc biologists thinking that notochordal cells are extremely important in the sense that they seemingly have a tremendous beneficial effect on the other chondrocytes that live in the nucleus. So some people argue that the notochordal cells disappear, but there's some data to suggest that they may not completely disappear. They just look differently in adults and may act as potential stem cells. But there's some studies that are coming out regarding this type of new paradigm shift. And then there's cartilage end plates, as has been already pointed out, and these behave somewhat like hyaline cartilage, but not really. And not much has been studied with this cartilage end plate, actually. People think it behaves like hyaline articular cartilage, but probably isn't. There's probably different phenotypic expressions. And then some people argue that this cartilage end plate also is a site where the cells can migrate into the disc itself, and Howard Ahn's group in Chicago has shown some of that. The data is not 100% convincing, but on the other hand, it's food for thought. And then Randy showed that some of the discs can get innervated during degeneration, but the outer annulus, about 1 millimeter or 2, is innervated, but the majority of the entire disc is aneural. So it's the paradox and the irony of it is that the largest tissue that's aneural causes most problems in our human's body in terms of pain. And now there are some mechanoreceptors in the end plates and in the very outer annulus, but for the most part, the entire sea of the annulus and the disc within is aneural and avascular. So, word about the physiology. As I stated, it's avascular. So any of the cells that live inside the disc has to get its nutrients through the end plate just by sheer diffusion and osmotic gradients. And because there's no blood vessels, it's a tremendously harsh environment. So these cells have to learn to live in a very acidic and very hypoxic environment. When you're a newborn, you do have some microvascular blood vessel penetrating the end plate and getting into the disc, but by the time you're an adult, it just becomes totally avascular. So most of the nutrients gets through the bony end plate through pores and through osmosis more than anything else. And then so many of the cells in the center actually have to live in a desert whereby the glucose and oxygen and various lactate or waste products have to get out, but for them to get in, it has to traverse almost 8-9 millimeters in distance. So it's quite a challenge for these cells. And again, it's just due to these little bony end plate holes, more or less, that you see in high resolution CT scans. And then sooner or later, when various glucose and oxygen gets in there, it can diffuse through. But again, as I stated, it's a harsh environment. Jill Urban has shown beautifully that it is all about the nutrition of the disc, and that's perhaps one of the reasons why we regenerative pundits feel that it may not be worthwhile, because whatever we put in the center of the disc has to survive, and with such a poor nutritional environment, it may be difficult, and there's truth in that. So you see lactic being very high in the center, oxygen, glucose being very low. But again, these cells have evolved over billions of years to survive in these environments, and they do the best they can, but probably sometimes they fail. And then, as Randy stated, there's mechanical compression. So these cells are not living in a non-stressed environment. There's adequate data to support certain loading is good for the disc, some loading is bad for the disc. So all the cells within the disc experience tensile or compressive loading or in combination. And again, the microbiomechanics is extremely intricate and very difficult, because there's some areas when you actually load the spine, the cells see tension. Other cells in the center sees compressive loading. So how that interplays with the disc biology is being studied actively, particularly in the last 10 years. Gwen Soa in our lab is actually looking at this, and she's noted in vitro at least, there's favorable loading environments to these disc cells, and then there's unfavorable loading environments. So if you overload the disc cell in certain frequency of tensile loading or compressive loading, it actually causes a catabolic breakdown, and you can actually see gene upregulation or downregulation of matrix products. And likewise, with certain favorable loading schemes, you can actually have a beneficial effect. So, finding the fine mix will be what's the challenge for the future. And if you start talking about inflammation and how it interacts with the mechanical loading, it's all intricately interwoven with each other. So if you have certain mechanical loading schemes on the disc, and then there's certain cytokine inflammation stimuli, the cell behaves differently. So it can augment or it can be detrimental depending on the different loading scheme as well as the different inflammatory stimulus. And it's thought that these cells behave differently just due to various mechanisms, and there's a lot of researchers that are looking into this now. The cells, how they form these microtubules, actin microfilaments, and when the cells get stretched, they behave differently and produce different inflammatory cytokines, and also produce different matrix proteins depending on the various external loading. Now let's talk a little bit about the pathophysiology, because this is now going into the painful disc, as we pointed out, but why the disc seems to degenerate. Is it just an aging process? And should we call aging a pathologic process? That's been in debate. If you start looking up the definition of what degeneration is with aging, it almost sounds the same. So we do know that everybody undergoes degeneration. We all age. There's no way we cannot age. But if you start looking at the pathomechanisms of what's happening to the disc during the aging process versus a true pathologic situation where the disc fails and degenerates progressively, it's very interesting, actually. So Dr. Eyre showed you that the disc can undergo glycation, deposition, but by and large, if you look at this quote-unquote semi-normal disc, an MRI may not look too terrible, but compared to a young disc, there's difference. So what the difference is that this disc has seen 50 more years of life. And so why is that? Is the aging process something different than a true degenerative process? So it's very interesting. I'm going to digress for 5 minutes to show you a little interesting experiment we've been doing on these little mice. If you look at the whole aging scenario, then the researchers that do the basic mechanisms of why we age, what is the fundamental reason why we turn gray, hair turn gray, we lose, we get wrinkles and we degenerate more or less. But why is that happening? Well, there's a body of evidence that shows that we age because our DNA gets damaged constantly. Every day we go out in the sun, we get damaged. Every time we ingest food, there's chemical ingestion of various toxins. We get exposed to oxygen radicals, and it puts little nicks in the DNA. And there's a clinical syndrome, of course, progeria, where it's been well described, all these different syndromes, and these patients age very rapidly. And it was not really understood why that they aged, but they had a short lifespan, and that was a problem. But ultimately, the final conclusion was that these patients with progeria age because their DNA just gets damaged rapidly, and they don't have mechanisms to repair it. So it turns out that all of us have a certain amount of endonucleases, which are these little enzymes that float around in our cytoplasm that identifies damaged DNA. So it goes around, cleans it up. So these endonucleases identify little DNA due to ultraviolet radiation and goes in there and just cleans up and reinserts a normal nucleotide sequence. So if you have a lot of these endonucleases, it could be the reason why some people age better than others. And it turns out that if you develop a mouse model of a endonuclease deficient sort of knockout mouse. Now you can't remove all of it because it's fatal, but you can remove a certain percentage, 10, 20, 30% of the endonucleases. And then if you do that, what happens is that these mice age very rapidly. So this is when they're born, they're normal, but with a certain amount of endonuclease has been knocked out, the mice just progressively age. And they look very old very quickly. These mice die in about 30-40% lifespan. This is the model of progeria that one of our researchers in Pittsburgh was investigating. And actually, when we saw this, we said, well, this is great for our aging mechanism in the disc. So we happened to take these discs and look at it. But these mice have all the classic signs of rapid progeria, very rapid aging. So because we have these models in Pittsburgh, we took some of their disc samples, and just the aging phenomenon alone in these ERCC minus delta mice shows that the disc is not producing as much matrix. And pathomechanism is probably DNA damage, and then ultimately the cells just don't make as much matrix proteins. And again, this is not really a severe degeneration, it's just pure aging. And probably the reason why the disc in a 60-year-old looks different than a disc in a 20-year-old. And then when you do these assays, you see about half the amount of GAGs being produced. Now, it's thought amongst the aging people that this whole aging phenomenon with DNA damage, it's a lot of complexities to it, but it's probably mediated by this NF-kappa beta transcription factor. It's a very ubiquitous transcription factor, but needless to say, it is a major component of this aging process if you talk to the aging gurus. So in this model of aging, if you can block this cascade of the NF-kappa beta activation due to whatever stimulus there is, with a, can't see this, but this NBD peptide, and just block this NF-kappa beta cascade of events that occurs with this NF-kappa beta inhibitory peptide. It's very interesting, and this was devised by Paul Robbins at Pittsburgh. You can take these mice that are progeric and rapidly aging and almost have a different phenotype. They have the same endonuclease deficiency, but if you block the NF-kappa beta cascade, you can actually delay aging, which is quite remarkable. And so you can see that all the phenotypic, you can't clean up everything, but you can get substantial lessening of the effects of the aging process in these mice. So there may be a cure for aging after all. So, and then if you look at their disc, you can see this with this NBD, just inhibiting the NF-kappa beta cascade, you build up the matrix again compared to the wild type, and then comparing it to the deficient mice. So moving forward with more or less these human situation, in the human situation, we're aging all the time, and then there are other pathologic components to the disc that occurs. And you see all these fissures forming, and there's no doubt there's mechanical environmental loading that occurs that causes this. There's these cells that start becoming dysmorphic. They form clusters, very much like articular cartilage. As the disc is being stressed more, the cells try to revive themselves in many ways. There's all these cluster formation that occurs, but ultimately there's an imbalance. The matrix metalloproteinases that these cells produce actually, in a way, break down the matrix proteins. And why it's doing that, obviously, is still not 100% understood, but probably due to the subacute inflammation. And then these proteinases, aggregate-kinases, MMP-generated genes that are produced by these cells probably act to make the disc degenerate further. Now, interestingly, as the disc is making these bad matrix enzyme proteins, it also tries to revive itself or try to balance it by producing these tissue inhibitor matrix proteinases. So it's a balance. It's a constant battle within the disc as what wins, the disc matrix breakdown or matrix build up. And then there's always these inflammatory cytokines that have been shown in the disc that's degenerating. And these inflammatory cytokines like TNF-alpha and interleukin-1s cause the disc cells to actually produce these various proteinases that may be catabolic. So again, this is the paradigm that we've used to devise potential treatment options, and it is a balance because there's various catabolic enzymes that have been well studied, and there's various anabolic cytokines that have been studied, and then when there is an imbalance, there is a tipping of the scale, and then probably is the paradigm why the disc starts degenerating. Now, there's all different mechanisms. We've talked about genetics, and that's very, very important. And, Dr. Eyre has shown you that there's vitamin D receptor, collagen IX, various things have been reported, but none of them have perfect association. So it, no doubt, is multifactorial. Genetics plays an important role, but the epigenetics and environmental factors and everything else comes into play as well. And I'll just finish off with the fact that we've already seen there's some discs that are not painful, some discs are painful, and the paradigm for that is that as the disc starts degenerating with the aging process or due to whatever pathophysiologic mechanisms at play, things start going bad. But many of us have disc degeneration that's not painful. But there are some of us that, for whatever reason, develop pain, and it could be due to the end plate, it could be due to other factors, but we seemingly are focusing on the disc. And if you take these degenerated discs, there are ingrowth of these various neovascularized tissue that comes into the disc and ultimately may be what causes the problem. And it may not be 100%, but people have shown over and over again in surgical disc specimens that are taken out for patients that have severe discogenic back pain, there's a significant ingrowth of granulation tissue into the disc and a lot of vascular tissue. And with influx of vascular tissue, there's various cytokines that get upregulated, and then you see neo-innervation of the disc itself. And so people surmise that this is what causes people with discogenic pain to have actual pain. And there's always, and as the disc is going through this inflammatory cascade, it's producing a lot of different sort of inflammatory cytokines as the matrix biology is being affected. And secondarily, if you have sensory neural endings growing into the disc, that is thought to be why these patients get the pain, because the sensory nerve endings has to undergo some inflammatory and mechanical loading to elicit a pain response. And so the general model, there's all kinds of pain people looking into this in the dorsal root ganglion, and you have various A delta and C-type fibers in the dorsal root ganglion that gets stimulated, and various cytokines that agitate the nerves start causing pain stimulation to occur. And then within the dorsal root ganglion, there's a lot of different growth factors that also mediate that response. And then finally, you don't experience pain unless it gets to the brain. And we know chronic pain is different than acute pain. And whatever happens up here, those patients with chronic pain are different animals than people with acute pain. So there's a whole area of research going on in here, especially when it comes to low back pain, since it causes so much morbidity in our patients. So in conclusion, I just given you a whirlwind tour of this complex nature of this disc, and aging probably plays a role, no doubt, but painful degeneration is very common. And a comprehensive cure, as a surgeon, I'm a little distraught at the fact that we can't cure these patients. No matter how much we try to cure lower back pain with surgery, we just can't do it. And I don't try. And so better understanding of molecular mechanisms may lead to novel biologic solutions. And I'll show you some in the afternoon, some of the genetic research we've been doing. Thank you.
Thank you. Well, good morning, and an additional welcome to Seattle. I'm David Eyre. I'm actually a PhD biochemist, so I'm going to kick this off with some basic science. I've been given a title that's a little challenging in a less than 15-minute talk: genetics of disc degeneration. But what I'd like to do is to use it as an example, or use examples of risk association genes for lumbar disc degeneration as sort of entree, because I think as basic scientists and geneticists and biochemists, protein chemists, it's giving us clues about what mechanisms may be operating that drive rapidly progressive disc degeneration, lumbar disc degeneration, particularly in actually younger individuals, early onset discs. So in 15 minutes, I'm going to go through a lot of basic science. I'm going to give, I'm essentially a collagen chemist and spent a career looking at collagen chemistry in skeletal connective tissues. So I'll give you some background, first few slides. I'm not sure if you will eventually get a handout of the slides with the references to papers that I'll mention. But I think one of the first points to emphasize is that at the molecular level, the molecular anatomy of an intervertebral disc in terms of its collagen fabric, which is really what fails when we get tears and then progressive degenerative changes, is highly specific and evolved. It's not just a mix of collagens that we see in other tissues in the body, like articular cartilage, cartilages, bone, ligaments, etc. Each of the molecules that has gone together to make a disc has spent millions of years evolving its own special chemical modifications. And that, as a collagen chemist, I think is important to understand when those of you who are thinking about trying to engineer or regenerate a complex tissue such as a disc or articular cartilage. It's challenging, to say the least. I don't think we're ever going to engineer something that's the original developed fabric. The best perhaps that can happen is something can be induced that will replace it long enough like my metal and plastic hip, that hopefully it'll last me the rest of my life in terms of disc repair. Having said that, let me make another point about genetics of disc disease, of progressive lumbar disc disease. What we've learned from the basic science of other degenerative disorders, particularly osteoporosis and osteoarthritis over the last 10 to 15 years, is those disorders we know have a very strong genetic component, just like disc degeneration. Usually the geneticists will tell us it's like 70% of the risk is genetically involved, genetics are involved. Well, that's true for osteoporosis and osteoarthritis and disc degeneration. The geneticists who've done genome-wide association studies for the last decade, hoping that they would come up with not one gene, and clearly not single gene disorders, but maybe 3 or 4 genes that if you were unlucky and had those variants or polymorphisms in your genetic makeup, in your DNA, that you were certain to sort of get rapidly progressive degeneration. In fact, what they found is there's a lot of very weak association genes with odds ratios in the population, of 1.1, 1.2, meaning you've just got that slightly increased odds ratio of, say, osteoporosis or osteoarthritis. Essentially, disc degeneration is the same. It's a very strong genetic component, 70%, but there aren't going to be genes that say you'll definitely get degenerate discs. So I want to give an example of what's been found over the last decade or so about genes associated with risk of degenerative disc disease. And I'm going to focus right in and use it as an example to give some background information on what we know about the structure of disc collagen particularly, which is the main fabric that's unusual. And I'm going to focus on, is this the pointer? Yeah. I'm really going to, we know all of these genes that encode these proteins have been shown in genetic studies to be associated with progressive disc degeneration. I'm going to focus on these two. Collagen IX, and I'll give a summary. Collagen IX is one of the so-called minor collagens, meaning it's minor in content, but it's actually major in its importance of setting up the structure of a cartilage tissue that's based on type 2 collagen. But aggrecan, proteoglycan, matrix metalloproteinase 3, vitamin D receptor, IL-1, all of these have come up in genetic association studies, have been associated with, mechanism unknown of course, but associated with more rapid disc degeneration, particularly lumbar disc is the clinical outcome that's been looked at. So again, background. We all know that the fabric of the collagen that makes up an intervertebral disc, the annulus fibrosus, the nucleus pulposus, is highly complex. I tend to think of a disc in gross terms. It's a highly evolved, specialized ligament in terms of its functionality. And it combines features of, say, articular cartilage at the molecular level and ligament. Collagen type 1, type 2, we found decades ago that there's a gradient going from pure collagen 1-like ligament tissue at the outer annulus to pure collagen type 2, a gel-like in the young developing disc, which actually molecularly mimics the molecules we find in vitreous in the eye. And there's a commonality between something like the collagen gel of a vitreous in the eye and the nucleus of the disc. We know with aging that we get all sorts of aging pigments, lipid oxidation products, glycation products that give this what used to be called lipofuscin, which is a mixture of sort of aging products that change the disc. But the real changes are sort of dings and tears and rips and the things that we at the anatomical level would call degenerative changes. Now, just to go into the summary of what's unique about, just for example, the type 2 collagen fibrils that are in the nucleus, but also as a gradient decreasingly through the annulus. If we could look with a super high-powered microscope, more than just a transmission EM, at the type 2 collagen fibrils, you would see, and this has been worked out over a decade or two from molecular studies, that those fibrils aren't pure polymers of type 2 collagen. They're actually built on this template in yellow of collagen 11. Collagen 11 is a family member of collagen 511, which really goes back in evolution probably 200-300 million years into invertebrates. This was the collagen of primitive organisms, but we still have it. It's the template that our bulk collagens that make ligaments, bone, tendons, etc., is built on. It's essential. It's about 3% of the collagen, say in an adult disc, but it's essential to make a new fibril. The red molecules are what I'm going to focus on now, where the polymorphisms that are associated with risk of disc degeneration occur. I don't know where that came from. Thank you. The collagen IX has evolved purely to interact with the surface of collagen II. In a sense, it's a template for the fibril network, we think. And again, over many years, we've shown that, I'm not sure why this keeps coming on. I'm not touching anything. Anyway, it covalently bonds to the surface of type 2 collagen, and we think that these bits that stick out form physical bonds with the proteoglycans and give strength or functionality to the matrix as a whole. But this part is covalently bonded to the type 2 collagen. Why, we don't know, but we presume it's because it's some kind of mechanical strength at the molecular fibril level. And you'll see why I'm getting at this in a minute. Again, just even more detail, if we look at this polymer of type 2 collagen, say, in articular cartilage of joints, and we look in fine detail at the structures, it turns out that each one of these three components, the bulk collagen 2, the collagen 11 template, and the collagen 9 on the surface, each have evolved different features. For example, the collagen 9 in the disc, nucleus and annulus, lacks a protein domain here that interacts with proteoglycans, NC4 domain. The gene is expressed and spliced differently. Similarly, the collagen 11 template of articular cartilage has a different chain composition in the disc. Again, this is the template for building the type 2 collagen. I'm just trying to get across to you at the molecular level, the devil is in the details. Disc type 2 collagen fabric that interweaves the thick collagen fibers of the annulus is a different molecular structure, and the cells control it differently. Collagen 9, just to summarize the properties, it's called a facet collagen, fibril-associated collagen with an interrupted triple helix. And it's a member of a family of collagens called facets. There's probably 5 or 6 of them. There are 30 collagen genes, 30 collagen types in the human body, all doing different things, like similar to the ones I'm showing. Facets modify the surface of fibrillar collagens throughout the body. Collagen 9 is peculiar in that it now only is made to cross-link to the surface of collagen 2, wherever collagen 2 occurs, even in the vitreous of the eye. We know that single gene mutations that cause disease, true mutations that are very rare, in each of the 3 genes, they call them for the chains alpha-1,9, alpha-2,9, alpha-3,9, the genes are Col9A1, A2, A3. Mutations in all 3 genes cause chondrodystrophies of various kinds. Mild metaphyseal dysplasia, for example, is caused by mutations in 2 of these genes, which gave a clue to the fact that maybe polymorphisms that are much more common in the human genome in different populations would be risk factors. The candidate gene approach for looking for genetic risk of disc degeneration. And here, a Finnish geneticist, Leena Alacoco, just over a decade ago, published a series of papers, but these 2 sort of hallmark papers came out in JAMA and Science over a decade ago that showed polymorphisms that were common in Col9A2 and Col9, this one's Col9A3, was associated with clinical sciatica disc degenerative disease as they looked at it. Their clinical outcomes were pretty broad. But again, to summarize, in the last decade, and if you go into the literature and are interested in this, there's a whole series of studies now in Japan, one out this last month in a large Indian patient population, that these are true associations. And I'll try and, in the last few minutes, just tell you where we've gone with this and try and summarize what it may be telling us about possible mechanisms for why you would have an increased risk of progressive, say, lumbar disc degeneration in reaction to the environment. Okay, so the two polymorphisms in question from myself, a protein chemist perspective, what's interesting about them is they're single SNPs, single nucleotide polymorphisms that change an amino acid in yellow here. Collagen is a GlyXY repeat. Tryptophan, W, never occurs in collagen. It sort of messes up the triple helical structure a little bit, so you don't find it. But the polymorphisms in question encode a tryptophan. The single base change changes what in most alleles in most people would be an arginine or a glutamine, these are the single amino acid codes, to a tryptophan. Now, in two different genes on two different chromosomes, this is where Col9A2 and Col9A3 occur, it couldn't have been a haplotype association, meaning just a link to another gene that was really causing the risk. So to have both being tryptophans indicated it was probably some kind of protein effect, expression effect of protein. And again, to cut a very long story short, but this is published, Yoshito Matsui, when he was in our lab here at the UW, when that paper came out, or those two papers from Alacoco, we got fetal human cartilage specimens from, maybe 50 or 100 samples and genotyped them, and then looked to see if the protein that was encoded with the tryptophan was expressed. And again, to cut a long story short, it's published. We could find in fetal cartilage the collagen IX associated with type 2 collagen became cross-linked. It was in the matrix. It seemed to be working okay. It got chewed up in the cell and wasn't used, which was the other possibility. Now, another important point is the frequency of these allelic variations, as they're called, in Asians is very high. The TRP2 allele, meaning W at that position, in one of the two alleles that you have for the gene product, 20% of Asians, this is Chinese, Japanese, Indian Asians, all have about 20% of individuals would have one allele, and many have two alleles, so they're homozygous for this tryptophan, and they have more severe disease, which you can find in the literature. The other allele has a different frequency. In Asian population, this was from a Ken Chung, Danny Chan study in Hong Kong, South Chinese, 0%, 1,000, I think 800 odd patients they screened genetically. None had this TRP3 allele. 20% had the TRP2. 5% of Caucasians have this allele, and only maybe 2% have that. So there are ethnic differences in the frequency in the population, and the incidence of disc degeneration in those with the triple alleles varies between racial groups as well, so clearly other factors involved. Again, I'm not going to be a couple of minutes. What we did, and again this is published, Yoshito Matsui again, in collaboration with Sohail Mercer here at Harborview when he was here in orthopedic surgery, we designed a prospective case control study of surgical patients who had spine surgery where we could get disc tissue, and we could also genotype the patients, an approved study. Bottom line, it's published in JBJS 2004. What it showed was that those individuals, and about 11 or 12, had a tryptophan allele that we had tissue from were genotyped. The statistics showed that the odds ratio was 18.9, that if they had the tryptophan allele, they would be in the severest end of 5 sort of grades of severity of disease. So spinal stenosis with spondylolisthesis was the most severe end. and 8 of the, I think it was the 11 who had tryptophanes were in that. So it indicated the data in summary that the association was risk of rapid progression, but not necessarily incidence of disease. Again, that spawned another study from the disc samples that we had surgical disc tissue. We could then look to see, is there anything special about collagen IX in that disc tissue in the patients that had progressive disc degeneration? Again, published, too complex a slide, but here's the reference, Su et al. Spine, February of this year. The bottom line summaries of these data were that surprisingly in normal adult human disc tissue, there's hardly any collagen IX. We had to find it. It was like the 0.0001%. It's gone once the disc has matured. But it comes back at focal sites where injury has occurred. It doesn't matter whether you've got the tryptophan polymorphisms or not. But what it tells us is that the repair response of injured tissue, when new collagen is laid down, collagen IX is expressed, re-expressed from decades after it had disappeared. One possibility is it's driving inflammation, perhaps, so that degeneration progresses faster. And this is summarized here. What we could find no IX in control disc tissue from fracture cases, auto injury, etc., by immunohistostaining or direct sensitive biochemical methods. And we only saw IX deposited in sites of injury. So we suspect that the expression is a repair response at local sites. And that may promote enhanced and progressive inflammatory changes. So this two sort of, and this is a slide acknowledging all the people in my basic research lab over at the UW who've helped with these studies over the years. But there are two possible overall mechanisms. If you inherit one of these alleles, it may be that the fabric of the disc is in some way, we know the protein would be in there from our studies on fetal cartilage, but it may be just misassembled ever so slightly or maybe deficient in quantity, so that those individuals that, which all of us do, go on to start developing tears and internal injuries in discs are at higher risk of developing a tear or injury because the fabric is not quite correct. But also, it's not and or, it could be both. Once the collagen IX with this abnormal allele is expressed at the protein level again, perhaps it attracts macrophages, etc., to drive maybe an innate immune response to the repairing tissue that's recognized as foreign, and that could drive inflammation. That's highly speculative, but it's an example of how basic genetics is giving us clues to processes that may be accelerating disc degeneration. And that's all I have to say. I'm sorry I've gone on too long. Thank you for your attention.
I will talk to you about the kinematics of degenerative disc disease. This is a very interesting topic. And if you look at the different terms that have been defined or used for degenerative disc disease, you'll see that we'll have a number of different descriptions: discosis, internal disc disruption, discogenic pain, disc degeneration, segmental instability. Whenever that happens, it already tells you that there's a little bit of a problem, that probably different people have a completely different understanding of what this really means. And I have to make a confession. In preparation for this presentation, I work a lot with Eyre Spine, and we have a database of presentations that have been collected for 10 years or so. So there are hundreds and hundreds of presentations that have been done by various spine professionals, and I was trying to cheat. I was going through that because I have access to that database. I was trying to find some kind of template presentation, and there was nothing there. So for 10 years, spine surgeons and spine professionals have been talking about all types of degenerative spine problems, but nobody really has talked about the kinematics of degenerative disc disease, or really what happens over time when you look at degenerative disc disease. So I found that very interesting. I want to talk about the classification systems that have been used for this chain of degenerative disc disease, and really what happens in patients who have these problems. This has been cited before by various authors, and one of the classification systems that has become, I think, fairly acknowledged and popular is a three-stage system where we look at the early stages of degenerative disc disease within the first 20 years of life, and then 20 to 60 years is stage 2. We develop a disc protrusion, disc prolapse, and finally, in the more aging population, 60 years plus, where you have ankylosing and collapse of the disc space. And there are very distinctive changes that occur in those different stages, and I'll try to kind of guide you through those changes because they're very, very interesting. Now, this I just put in there to indicate that obviously this is a very multifactorial picture, and various other speakers today will talk about the genetics, the molecular changes, and so forth. I would really try to focus more on the pathological and the structural changes that we can see in degenerative disc disease. Now, stage 1, again, is the stage that happens pretty much from birth until we're like 10, 15, 20 years old. What happens is that there's a regression of the intradiscal vessels. So the nutrient supply to the disc depends more on diffusion as we go through those years in life. Initially, little children, obviously, the body weight is very low. There's a high activity level, which is very favorable because that causes a lot of movement-dependent fluctuation of nutrients and water in and out the disc. As we then get older, as we start going to school, we take up jobs, we get heavier. There's an increasing mechanical component and axial loading that exerts some disfavorable impact on the discs. There's weakening of the annulus fibrosus lamellae, which is partially induced by the problems with nutrition. And then we start to see bulging of the discs, and sometimes even already pain in these young people. Now, this is just a slide that kind of demonstrates that impact of motion and loading in the early stages, where you have compression of the discs, where fluid and waste products are being pushed out of the disc, and then as you're relaxing towards the end of the day, that goes back into the disc together with nutrients. So that's that's really what we're looking for. That's kind of a sponge-type mechanism where you actually get nutritional supply to the disc by compressing and then really by a separation of the end plates, and therefore by diffusion and mechanical fluid shifts. Now, if you look at the lamellar structure of the annulus fibrosus, this is obviously a very complex anatomical situation, and you can see that as long as the lamella of the annulus fibrosus are intact, this looks like a structure that will be able to resist tremendous forces and really provide an encasement and a very stable situation for the disc. And as that changes with the nutrition becoming more problematic, any type of change in this lamellar structure may actually impact the stability of the disc and may lead to fissures, ruptures, and so forth. And that's actually what happens. Now, if you look at the forces that the spine is kind of exposed to over the course of a day, these were studies that were done actually in humans where they implanted little sensors into the disc space. You can see that the most favorable situation is when you're lying flat, recumbent sitting position. But then if you stand up, standing up, bending, and lifting is really the worst that you can do. That really impacts the greatest forces on the disc. So the lamellar structure of the annulus fibrosus has to withstand all these situations. This slide is actually from one of those studies where they implanted the sensors into the disc. I put this slide in for Jens because this gentleman here is lifting. That's probably what he did last night when he came home. He's taking the cases of beer and brings them up, right? So you shouldn't do that, Jens, because that's not good for your disc. But this is actually how they measured different body positions and how that affects your discs. Now, stage 2 is the stage when you're between 20 and roughly 60 years of age, and that is really the stage where a lot of the pathology happens and where we actually see a lot of these patients. Initially, the nucleus pulposus is still intact. However, we talked about the progressive weakening of the annulus fibrosus because of the nutritional problems. So we start seeing tears or little weakening in the annulus. Initially, there's actually increased pressure in the nucleus pulposus. There's increased osmotic pressure because of the breakdown of internal proteins. There's asymmetric axial loading, and we start seeing disc herniations at that stage in life. And then as we progress over the years, there starts to happen a slow decrease in water and in collagen content and so forth. Now let's talk about the axial loading. What has been shown in degenerated discs is that if you apply compression and bending, for example, you will not get in degenerated discs that kind of smooth equilibrium of loading that you see in the normal disc. But in a degenerated disc, the forces will be unevenly distributed over the surface of the disc because of the degeneration. And that can obviously then result in further mechanical injury to the disc and to the disc structures. And that has been shown to actually happen in a degenerated disc. What are the precipitating factors for disc prolapse during that stage 2 of degeneration? So the increased mechanical stress, for example, the uneven loading, the elevated trigger pressure of the disc, the loosening, or the overall loosening of disc tissue, the weakening of the annulus fibrosis, all these factors together can result in a situation where patients can actually have a disc herniation that may or may not become symptomatic and may require more treatment. So this is a pathology specimen here where you actually see the internal disruption in the disc herniation here. That's an extruded fragment, that's the same fragment 24 hours later, where you can actually see as it's in physiological saline solution at a normal temperature, at a 37 degrees temperature, what actually increases in size. And that is the reason why sometimes disc herniations can get bigger while once they're extruded, because they take off water, they start swelling, and then hopefully over time they will shrink down again. That's the annular fissure here, and you can see a herniated disc on this pathology specimen down there. Now, this just summarizes kind of the events that facilitate the disc rupture. Over time, the prevalence of annular tears goes up as we get older. The trigger pressure of the nucleus pulposus in the middle-aged population actually goes up, so the pressure inside the disc is higher, but the annulus fibrosis is weaker, and that's why we see most of the disc problems in the age group of 20, 30, 40 years of age. This is an annular tear seen in the operating room under the microscope. This is where a disc herniation actually happened, and that's sometimes what we're faced with as surgeons, that we remove a herniated disc and then we look at the disc space and we see this huge annular tear. The problem then is, or the challenge is always, is there any way to repair that? So what about instability? We always talk about instability and disc degeneration, and there are different ways of really classifying instability. But if you look at the structure of the annulus with the collagen fibers going up and down, as long as the annulus is healthy, it is very, very strong and very good at resisting twisting motion, translational motion, separation of the end plates, and it provides a lot of stability. You can imagine as these processes occur that I described, as there's weakening of the annulus fibrosis, that there can be a situation where you actually see some type of significant instability with spondylolisthesis and even movement that you can pick up on flexion extension films maybe. And that's what has been studied and has been described. This is a paper by Howard Arndt's group where they looked at 4 different stages of disc degeneration. They obtained MRI scans and flexion extension films in a large number of patients. And then they looked at the stability of the spine there, and they could see that initially when you're in stage 1 with very minimal degeneration, you have a relatively stable spine, but the movement then in the stage 2 phase, the instability actually increases, and then as you reach more extreme stages of degeneration, the spine really becomes more stable again. So there is indeed that 3-phase picture that I'm trying to explain here, where you see more instability in that middle phase, where we actually also see more disc herniations and discogenic problems in these patients. Now, the 3rd stage is really the stage where everything kind of settles down. There's increased dehydration, stiffening, calcification of the tissues. Some have called that the comfortable rigidity of the aging spine. Just to summarize, the etiology of degenerative disc disease is obviously multifactorial. We've heard a lot about the genetic predisposition. What I really focused on is the premature aging of bradytrophic tissues indicated by static mechanical forces due to upright posture, the increasing weight as we get older, the insufficient nutrient supply, and the weakening of the annulus fibrosis and the generation of tears or fissures that then result in more problems. Now, the pathogenesis, I'm not going to go through this because this is all pretty well known. But I want to just mention a few sentences about pain. What is the relationship between pain, low back pain, and the disc? And this is something that is obviously very important because we kind of assume that degeneration causes pain, but this is not always the case. Anatomically, we know that the annulus fibrosus is innervated from the dorsal root ganglion, and that is maybe the structure of why people who have irritation of the annulus fibrosus and the posterior longitudinal ligament experience pain. However, and the way that this has been thought of is that you have an annular injury, you have an inflammatory response, which creates some type of scarring, vascularization of the tissue, increased innervation of the posterior longitudinal ligament and the annulus fibrosus, and that in conjunction with abnormal loading then can result in pain. Now, we know, however, that there's no direct one-to-one relationship between disc degeneration and pain, and I think we always have to keep that in mind. We know that pressurizing the lumbar discs, which is something that we do, for example, with a discogram, can cause pain, and that has been studied as early as the 1940s. However, the other way around, there's no direct relationship between disc degeneration necessarily being associated with pain. So I just want to mention that because that is very important for those of us who treat these patients, that we don't make this assumption. 35% of healthy volunteers show significant disc degeneration. However, there's no direct relationship between disc degeneration and low back pain. There was just a paper out a few weeks ago looking at end plate lesions, and the authors here from Asia and from Finland bring up the interesting alternative hypothesis or additional hypothesis that a lot of the pain that we frequently associated with degenerative disc disease may actually be related to end plate changes and not necessarily to the disc itself. This was an autopsy study where they looked at end plate lesions such as Schmoll's nodes, fractures, erosions, calcification. And in these autopsy specimens, where they had a very good history of the back pain as experienced during their lifetime, this autopsy study showed a very close correlation between these end plate changes and the history of back pain. So I just brought that up because I think it's a very interesting thought, and we should certainly keep that in mind. This is another pathology specimen showing an end plate fracture here with protrusion of the disc material into the bony anatomy. Now, in terms of future therapy options, I think if you look at the whole cascade of events with degenerative disc disease, to me it becomes very obvious that there's probably not going to be a mechanical solution to the problem. The whole process is so complicated and involves so many different components of the anatomy, and there are so many stages that individuals go through that it would certainly make sense to look further into some kind of biological modulation or treatment strategy at various, depending on the stage of the degeneration of the disc and whether or not that is then obviously responsible for the patient's symptoms. Studies have been done looking at protein therapy, growth hormones, platelet-rich plasma. Now there are some really interesting, exciting results on juvenile chondrocytes that are being used to treat relatively early stages of disc degeneration, mesenchymal stem cells. Some work has been done, but not very successfully so, on gene therapy and then tissue engineering. So I think if you look at this really fascinating cascade of events, these biological treatment options become more and more interesting. Now, concluding, disc degeneration is an aberrant cell-mediated response to progressive structural failure. However, in itself, it's not necessarily pathological because we all experience that. It becomes pathological once patients actually experience pain that we can correlate to that particular degeneration. There are at least 3 phases that are relatively well described, and the etiology is multifactorial. A lot of it is genetic, but there's also certainly an important component of progressive mechanical changes, impaired disc nutrition, and so forth. Then there are other factors involved, obviously, that we'll talk about later, I'm sure. Overall, I think it's poorly understood. The question is, are there other adjacent structures, such as the end plates, that may actually be more important in a lot of patients? And then there are interesting future treatment options that we should keep our eyes open for. Thanks a lot for your attention.